Screen right now?
Yeah, and we're about to start here.
Okay.
All right. Hello, everyone, and welcome to phyloseminar.org. The current theme is tree inference from non-traditional data. Today, we'll hear from McKenna about inference of organismal differentiation using CRISPR arrays. If you want to ask a question, you can either tweet at phyloseminar or type your question in the live chat box to the right of the video on YouTube. As I mentioned, today's speaker is McKenna. Aaron is a postdoc in Jason Dury's lab at UW Genome Sciences, which is where he did his PhD. Before his PhD, Aaron spent almost a decade as a software engineer, first working on an NSA contract and then developing the GATK toolkit at the Broad. He's headed for a faculty job at Dartmouth now. Welcome, Aaron, and thanks for participating.
Yeah, thank you so much for having me, Eric. This is really fun, and I think I'm going to bring kind of a slightly different perspective than some of the previous talks. Mine will be a little bit more about how we get the data, but hopefully it's interesting to everybody. And I can't see questions come by, so feel free to interrupt me as questions come in. So as Eric said, I've been really interested in developing tools to reconstruct cellular lineage, and the tool we've been using is CRISPR. So I'm going to tell you a bit about that today. But first, it's good to kind of step back and ask why we're doing this. So if you go back 150 years, there's been this really open question about how we develop as an organism, how we go from a single cell all the way to the trillions, 37 trillion cells we have in our bodies, and how are those cells maintained, and how is this process conserved across organismal development. And this kind of problem is in some ways analogous to other problems that we see in phylogenetics. So for instance, how have people developed over time? How has society spread throughout the world? Or how does the pedigree develop within a family, right? How do we recover this information? And so the kind of central model for all of this, right, is we want to go from some data that we've collected, develop a model of how this data is related, and produce a relationship. And we tend to do this in the form of a tree. So if you're looking, say, at the tree of species here, we may collect data on a specific protein, look at how that protein evolves, build a model of that evolution, and use that model to produce a tree. In this case, the tree of all vertebrate life, or at least a sampling of vertebrate life. And this, of course, has been kind of the bread and butter of phylogenetics for the last century. And what the real problem that we were trying to tackle is that this model or this process doesn't really exist for development. So if you take that kind of same picture here of development, and if you go down one of these columns going from the kind of early stage of development to nearly a fully formed mouse or human here on the right-hand side, what model do we have of that process, of those cell divisions? How does that happen? And then how do we use that model to build the relationship between all of the cells within the body? And one course answer that's becoming more and more prevalent, and this is a plug to check out Miriam's talk last week, is to take something like single cell transcriptional data. So for every cell, you have some information about what genes are being expressed at that time, and build up a model of how that changes and use that to infer the relationships between individual cells. And much like in Miriam's talk last week, you can kind of build these trees of the relationship between cells over time. And one of the real questions with this technique is what range of development can we cover with this? This transcriptional data is pretty complex, and we don't really understand the relationships between the expression of one gene over time and all other genes. They're certainly not independent like they are, or like we assume in, say, the kind of protein evolution perspective. So how, what can be done about this? How can we infer the relationship over all of development? And what methods can we come up to solve this problem? And so the best way is to kind of look historically, what have people done? And of course, you don't have to look much farther than the Nobel Prize. In this case, Alston and colleagues. So what Solston and colleagues did was build up this kind of, I hate to use the term, but this phylogenetic tree of cell lineage. So they built a tree of the relationships of each cell in terms of cell divisions for the whole C. elegans. And you can kind of imagine this process over time where Solston, wheels into his desk every morning. And there's a great kind of write-up on this in one of the worm books of Solston wheeling into his desk every morning and doing this over and over again, recording each individual cell division over time and slowly building up this tree. And here's on the left are some of the sketches that Solston made in that process. And you can they actually have pictures of like kind of the scars on the floor his chair left as he wheeled back and forth, picking up worm after worm. And what him and his colleagues were able to do is build up this lineage tree of all cell divisions over time. And he has this great quote at one at the end of the papers that this report marks the completion of a project begun over 100 years ago, determining the entire cell lineage of a nematode. And I mean, this is just a phenomenal work, of course, that won the Nobel Prize. But how do we, how do we extend this project from the C. elegans, which is arguably a relatively simple multicellular life, to more complex cellular systems? And so it's important to note, what made this possible. First, C. elegans has a deterministic lineage. So every time Solston wheeled up to the chair, he was able to pick up a worm and see the exact same process happen time and time again. The worm is also transparent in their life cycle, so they could watch it with a light microscopy. They also had great advances in those microscopy systems, and they really leveraged that technology to build this lineage up. And of course, as I talked about before, just the persistence of collecting this data over a huge swath of C. elegans development. And I'm going to try to play this video. We'll see how this goes. And you can contrast this with more complex vertebrate life. So what we're looking at here is about 800 minutes of development in early zebrafish embryos. And each one of these colored dots is a cell migrating, and the colors here are just which perspective the cells are moving in. And you can see just the kind of enormous complexity of this process as cells move, divide, migrate, and reform intricate structures. And this, of course, is just an early phase of development. And here you could count the number of cells. As we move into, say, adult humans, those 37 trillion cells become a much, much larger problem. So how do we develop technologies that scale to this problem? And of course, you can look at development, but you can also look at other systems that we're interested in at the single cell level. So say the development of a tumor, right? The belief is that a single cell seeds this tumor, divides repeatedly, and leads to a very heterogeneous population of cells performing various tasks for the tumor. So how do we develop models of all of this? And of course, there are purely computational options, but we really wanted to set out to solve this problem both on the experimental side as well as the computational side by bringing kind of new tools in both domains. So what would we like a system like this to look like? Well, what's done currently is we label a cell, whether it be a fluorescent mark or even 100 years ago, labeling it with something like a vital dye. The cell divides, and its progeny go on to form different parts of different organs if we label early enough. So this yellow cell that we've labeled can go on to form cardiomyocytes in the heart, cells in the stomach here, and the lungs. So it can really lead to a diversity of different cell types. But the problem is that we're just labeling one cell at a time. So we need to know what stage of development to label, and we need to label every possible cellular lineage at the resolution that's important to us. And importantly, this is a destructive process. So we can't just watch this cell in a non-transparent animal. We have to destructively sample from the organ or tissue of interest. So that means that we'd have to slice at every time point that we're interested in and recover cells and assign this lineage. And of course, this is not news to this audience, but when you're talking about 37 trillion cells, the tree space that you can build from this is just enormously complex. I just have some examples here for very simple trees, but you can imagine this being 37,000 or 37 trillion cells. And it's just an enormous, enormous space that we're working with. So, and why do we want to do this, right? Well, so you can look back at that C. elegans lineage and we can ask what we've learned. And so much of what we know about cell death and its implications for cancer come from knowing the lineage of that model organism. There's really interesting examples in human development. For instance, the kind of cardiac development and the vasculature around the heart come from very, very diverse lineages. And there's implications in its construction on those lineages. So where, for instance, vasculature breaks, there's some evidence that at these diverse lineages where those come together, those junction points are the weakest points. And of course, as I said before, the kind of evolution of tumors is a really, really interesting space. And my time at the Broad was trying to do a lot of computational modeling for tumor development. And this really motivated me to come and do both experimental and computational tools. So today I'm going to tell you about the tools that we've developed based around CRISPR-Cas9 barcoding, its application to zebrafish, and its kind of the addition of single-cell information on top of that. So first, we wanted to set out to develop an experimental approach to label cells. And much like everything in the experimental world right now, we turned to CRISPR-Cas9. And we had a kind of very simple concept. So you can imagine that you want some way to record information in the genome that's independent of any kind of mutational signature that the cell is being exposed to. So what we imagined is that you could put a small barcode into the genome, and we'll represent that here as this white bar. And you can target CRISPR-Cas9 enzyme to places in the genome. And here we have 2 targets. And when CRISPR-Cas9 binds to those targets, it creates a double-stranded break in the DNA. And that has to be repaired by the cell for cell division to progress. So the repair process can be error-prone, and it can leave insertions or deletions. And for the rest of the talk, we'll have this blue be an insertion and this red be a deletion. So we can make these targeted insertions and deletions in a very specific part of the genome. So we can engineer in a cell a small piece of DNA that can be targeted by CRISPR. And what we can do then is, as CRISPR leaves edits over time, it starts to mark the lineage of individual cells. So you can imagine that we inject Cas9 into this first cell, and it targets our small little barcode here. And the progeny of this cell, the 2 daughter cells we have here, can take on different stochastic edits. So you imagine one here has a deletion in our barcode, and one has an insertion on the bottom. So this, of course, isn't much information at all. We need this to be much more densely packed. So you can imagine putting in many targets into the genome in this small little compact barcode. And here we have, I believe, 10. It should be 10 targets. And what you can do then is, as these 10 targets start to take on different insertions and deletions, you can differentiate individual lineages of cells. So if you look here on the right-hand side, we have an insertion at the, let's call this the 10th target, that we can use to group these 6 cells together into one clade. We then have these further deletions on top of that that delineate these subclades. And we can also infer then the historical editing pattern here. So we can say in this subclade, some progenitor of all of these cells had this single insertion at the 10th target and this deletion layered on top of that. And we can refine that again by adding this insertion here at the first this deletion at the first target. And we can really build up this lineage of individual cells. And one of the really nice things about this is that as we add more targets and as we add more cells, we get more resolution. So it really nicely scales with at least scales with the size of our sampling, which is really nice. And then it's not a big leap to imagine in this synthetic system that that first cell at the beginning of all of this is the single cell zygote that we all start as. And these cells are sampled from diverse organs and tissues. And of course, we have to come up with a name for this kind of thing, especially in the experimental realm. It's not a method if you don't have a name. And we came up with Gestalt for genome editing of synthetic target arrays for lineage tracing. And it's a little bit of an aside, but it's worth kind of saying what we're doing here. We capture all of this information from single cells, but we're not capturing it with the single cell transcriptome or other cellular data. What we're doing is actually tagging a single genome's worth of DNA with a unique identifier. We're PCRing that up, and we're collapsing that all down to a single representation for the cell. So we've labeled each cell's barcode, but we don't know much more about the cell. And I'll talk a little bit more about that in the single cell section. And what we do is then aggregate all that information into this barcode. We look at each of these targets, and you'll see here, if I can find my cursor, maybe not. You'll see here on the bottom, over each of these individual targets, then we call a particular event. So at these first two targets, they're labeled as none because they're, say, wild type. And the next three targets incorporate this 61 base pair deletion, and then again we have a wild type sequence. And I'll represent that in the rest of the talk as this colored barcode on the bottom, where we have white be the wild type sequence, red be deletions, and blue be insertions. So one of the kind of questions that we had to answer before we really started this project was, can we actually generate information content? Can we make a diverse editing pattern in this barcode? So we did something very simple. We put the barcode into human cells in cell culture, let them grow up for 7 days, added Cas9 before we let them grow up for 7 days, and we'll ask the question, can we see diverse editing pattern in our barcodes? And I'll just show you these plots because they'll pop up a lot in the talk. We want to see this indel diversity in a kind of compact form. So what we'll do is on the top, I'll show a histogram of all of the insertions and deletions over a barcode. And the red will be the deletions again, blue is insertions. And here you can see over our 10 target barcode, we see a lot of deletions at this first and third targets. And we see deletions that span, as you'll see, many of these targets. And below that, we'll have these white bars here, which represent the individual alleles. So in alleles here, we mean all cells that have the same editing pattern. So you'll see here we have 10 to the 5th wild type cells with no edits, and about an order of magnitude less with a single deletion at this first target. And what we'll see in this cell culture system is that we actually do get a pretty diverse editing pattern. This is looking at about 1,600 unique sequences. We're just showing the top 25 here. And in this design that I'm showing, there's certainly diversity, though it's kind of packed into this first few targets. And we can tune this editing diversity. So by choosing a different set of targets that are less active, we can really change this y-axis. So we go from about 50% edited at that first target down to about 1%. But what we do is we really, by turning down the editing rate, we make these targets more independent of each other. So there's not 2 times that Cas9 binds and drops out the intervening DNA. Instead, we get repairs at just single targets.
It might be worth mentioning, I mean, this task of going from the curve to these discrete set of alleles is not totally trivial. And it's sort of, in a sense, like the moral equivalent of multiple sequence lineup that we do in usual phylogenetics.
Sure. Yeah, no, and there's certainly both a lot of experimental details and a lot of kind of computational details that are being glossed over here. though they're a little less interesting in my mind. But yeah, no, it's certainly not a trivial problem. And I think one of the things we're seeing in single cell data too is that missing data is also a challenge, where we've lost, essentially lost the whole barcode in a process that we're not always completely sure of what happened. So, but no, that's a really good point. So we're pretty convinced at this point that we can make at least an information-rich barcode. And what can we do with it? So one of the things we just want to test, right? Can we make a synthetic system that in some ways mirrors development? It mirrors the cell lineage tree within an organism. And of course, we do this in cell culture because it's much, much easier to do than any model organism genetics. So what we can do is we can take an individual progenitor cell with the wild type barcode, take a single cell, expose it to a round of this CRISPR-Cas9 editing, sort that out into various single cell subpopulations, let that single cell grow up into another population, hit those individual populations with CRISPR-Cas9 editing, and then sample from those populations. And what we're really hoping to do here is to build up this tree, or build this tree that we, of course, we know, but we'd like to be able to infer this from the data. And so just to kind of zoom in here, what we're really doing is akin to a pedigree tree. We want to say, can we assign these daughter cells back to their parental line? And can we separate the individual parental lines from each other? And if we look at this, just one of these lines here, you can look at the ancestral population after that first round of editing. And you can see it has this really kind of defined edit at the first target. This is a 6 base pair deletion in the first target. And then after that second round of editing, what you can see is that we've really acquired information that allows us to differentiate both this daughter population from the ancestor, but also individual daughter populations from each other. So we're able to say this daughter comes from this ancestral population, and each of these daughter populations is unique by our barcode. So can we do this for that whole synthetic tree we set up? And it's an important aside for this group. We took kind of a simple approach to build up these trees. So we took that allelic representation I had before, where we call events at each of these individual targets. We took that and encoded it into a binary vector and fed that into camel local parsimony to build these trees. And so we took that final tree representation, and this is with mix. And one of the challenges here is that is, of course, builds a bifurcating tree. And in certain internal nodes, we don't really have information to differentiate one branch point from another. So we have these kind of artificial branch points that make it a binary tree. And so one of the last steps we do is to collapse this down. So if internal nodes don't have any information that differentiates them, we collapse it down into a multifurcating tree. And so what we're left with here is that kind of tree representation on the bottom.
Maybe it might be worth just reminding people what camel so-called parsimony is, because I had never heard about it before reading your paper. So
okay, sure. Yeah. So camel-local parsimony is a pretty simple parsimony model. It has a couple key assumptions. First, that, and I think most importantly, that we're only allowed to go from in our case, the unedited state to the edited state. So we can only go from a wild type target to an edited target. And this is a pretty key assumption for us, because there's certainly strong belief that once CRISPR is very specific, so that target sequence you put in, single nucleotide changes are enough to essentially ablate CRISPR's ability to edit that site. So as soon as it leaves an insertion or a deletion, that event is really locked in. And so we wanted our parsimony model in tree building to encompass that. And so camel-local parsimony requires that you can never revert the state of a change, right? So you can never go from the edited state back to the wild type state. So that was one of kind of the key assumptions that we wanted to use. And of course, the kind of state matrix here that we're allowed to build can be really large, and it's still tractable to run chemosocial parsimony for tens of thousands, or at least thousands of allelic representations.
Thanks.
So can we build this tree for all of the cells that we recovered in the synthetic lineage experiment? We can, which was really, really encouraging to us at the time. We weren't quite sure how this is going to work out. And as the title of the slide says, we're able to assign 99.7% of the cells, the daughter cells, back to their parental lineage. And of course, the asterisks should make you think really quick. And that's for this orange cell population here. And we're kind of cheating with that 99.7 number here because this orange population seems to have actually been two cells that were sorted into a well that made this specific population, one of which had this really kind of defining event in the population here on the far right, and another that was actually a wild type barcode that was free to take on whatever insertion or deletion pattern it wanted. So that kind of second population of cells is sprinkled throughout this tree. So that's certainly a caveat of this, is that we're really tied to the diversity and the timing of these CRISPR-Cas9 edits. But again, it was really encouraging to us that we could build these trees of cellular lineage just from this simple barcode. So what do we do with this, right? That's a really nice little kind of toy example, and we were really excited that this worked. But you want to kind of take this into real development. And we chose the zebrafish. And why did we choose the zebrafish? Well, there's lots of really good reasons. Zebrafish is a complex organism. It has many of the organ systems akin to human development. And it's really one of the principal model organisms for vertebrate development, especially early embryogenesis, as you can see here on the left. A nice kind of technical reason to choose zebrafish is that you can actually just inject reagents right into the zygote. I had no previous experience with zebrafish biology, and even I was able to give it a try. So it's really amenable to our approach here. And lastly, had a really great collaborator, James Gagnon, who now has his lab at the University of Utah. At the time, he was a postdoc in the Shear lab at Harvard, which is really one of the foremount labs for zebrafish development. So the first thing that we did was just inject all of these reagents into zebrafish single cell zygotes. And we wanted to see, is this going to perturb development, right? You can imagine you're putting a lot of genome editing reagents into a single cell and hoping that it looks normal. And thankfully, they really did look normal. You can see the examples here on the left, and you certainly don't have to be a zebrafish biologist to appreciate that they look pretty much the same. In 38 of our 39 injected zebrafish on the bottom, they really looked like they had all the bits and pieces that we would expect from a zebrafish embryo. And that could mean that nothing is working, of course. But when we PCR'd out the barcode from these whole zebrafish, what you can see is that on the uninjected side here in the middle, we get a really solid band representing our intact barcode. But when we looked in these edited zebrafish, you see this smear, right, which represents various insertions and deletions, especially deletions, popping up in our data. So we were really encouraged to kind of take this further. And so we set up this kind of large experiment here where we took a fish with the Gestalt barcode integrated, crossed it to wild type fish, harvested those embryos, and harvested those zygotes, injected them with Cas9 and the guides, and then sampled throughout development. And what we wanted to see then is, how does this barcode evolve over time? Does it continually record signals of development? And kind of when does it stop? And so one of the first things we did was just grab one of these 30-hour zebrafish, PCR it up, and look at the editing pattern that we saw. And one of the differences between here and the cell culture data is that we actually have guides for each of these targets in there. And that really paid off. We see a much larger diversity of editing outcomes, as you can see in the plot below, that really gave us hope that we're recording a lot of information in these embryos. So we then did this for a large number of embryos. I think it's 43 embryos that we did this for. And we wanted to know how many cells did we capture? And by cells, I mean how many unique molecules of our barcode did we capture? And you can see here in one of our 72-hour fish, we're capturing just over 22,000 or so. And throughout development, you can see that this kind of scales with the number of cells at that point. So at 4.3 hours post-fertilization, we expect there to be about 2,000 cells, and we're capturing upwards of 1,000 of them in this assay. And this scales out to some of the better fish at 30 hours, having over 20,000 cells. And so we're capturing a lot of barcodes. Do they look good? Well, if we look at the number of sites edited, we're seeing upwards of 7, 8 sites edited in each of these barcodes. So we're really recording a lot of information. And if we reduce that down to the kind of unique editing patterns that we see, so take the number of sites edited, collapse all of that down to a allele over all of the targets, and look for unique alleles. And this really scales nicely. We see hundreds of unique editing patterns in the 4.3 hour fish, up to thousands in the 30 and 72 hour. So we are recording more information over developmental time, which is great. That's what we were hoping the system would do. And we can use that same kind of system that we used before with Camm-Sokal parsimony to take these alleles and reconstruct a tree of cellular development. And we did this for we did this for all of the fish that we captured. So this is actually 45, embryos over development. And you can see the diversity of kind of information richness that we get back. Some trees are much larger than others, depending on the kind of random insertions and deletion rates that we get. And of course the recovery. So you can see in the upper right-hand corner, we get one fish that has, I don't know, 40 barcodes or something like that, a pretty limited set. But some of them we did really well. And I just want to show you one of the 30-hour fish where we did a whole lot better. This is capturing thousands of cells, and you really get upwards of 7 individual cell divisions in any of these trees, and really rich diversity of potential lineages. And you can zoom in here and kind of see how this works. So this lineage here has this really defining deletion here at the 5th target that spans the full sublineage. And then you can see these additional deletions that delineate subclades. For instance, that larger deletion at the 7th target in the upper half, or that insertion in the kind of middle of the tree. So we can really build this richness into our barcodes and then recover it and build pretty informative trees with it. So this is interesting, but we really have nothing. We have no information about any of those individual cells, and none of this was done at the single cell level with, say, single cell RNA sequencing. But what we did have is we did have organs of adult fish that we could dissect out. So we let some of these injected embryos grow up to adult zebrafish and dissected out individual organs. For instance, the brain, the eyes, different parts of the intestine, the blood, heart. And one of the nice things about this system is that the integration marker is actually a marker of cardiomyocyte development. So cardiomyocytes will come up as GFP positive, and we can actually flow sort them out. So we have now at least some annotation for any of these barcodes that we recover. We can assign them back to one of these organ types. And so we dissected 2 adult zebrafish. I'll just talk about one of them here today. but we're able to recover about 200,000 cells, or barcodes for those cells from this fish, spanning these 11 different organ types. And if you look at the overall diversity that we get from that 200,000 cells, we're getting about 1,700 unique editing outcomes. And of course, we would love that to be higher. We would love that number to match the cellular diversity that we have, but this is certainly a great place to start. So what can we do with that? Well, first, let's kind of look at the editing patterns that we get in these individual organs. So here you can look at the blood, and you can see the kind of a slightly different editing pattern than we saw in some of the embryos. So the first, it's worth noting here that the far right plot is log scaled. And so what we're seeing is that the allelic diversity is pretty constricted here. So the first 5 alleles here make up over 99% of the blood. And other organ types are certainly more diverse. If we look at the brain, for instance, we're seeing a much more diverse editing pattern here and a kind of greater number of unique alleles. And you can also see that the blood is, of these same blood alleles are showing up in the brain. And of course, this is something that we should have expected but didn't. In some of the first fish we processed, we didn't sample blood. And that kind of signal is everywhere because it's really hard to get blood out of an organ. And so, but that, of course, kind of leads an interesting question. If we look at that blood proportion in all of the organs, so those 5 alleles that make up 99% of the blood, you can actually see those in every other population. And you can see them in the same rank order that we saw them in the blood. So these blood lineages are equally spread out in all of these organs, and we're able to recover those. The only population that we don't see them in is cardiomyocytes. And this was a great sanity check. We sorted cardiomyocytes based on this GFP signal, and they should be very, very, very pure for cardiomyocytes. And we're really depleting any blood signal. So both our experimental protocol is working, but also if you think about the relationship, cardiomyocytes and blood are pretty distally related. And we're really sharing no unique alleles between cardiomyocytes and blood. So we're at least capturing information past the divergence of blood and cardiomyocytes, which is really encouraging. And you can look at this kind of sharing between all kinds of different organ combinations. And what you see tracks really well with development. So for instance, if you look in the lower right-hand corner, the brain and the eyes share some allelic signatures. But if you look at something like the brain and heart, for instance, you see almost no overlap. So we are at least tracking somewhat with the developmental signals that we would expect to see. And one of the kind of really interesting things here is that we do see those top alleles really dominating individual organs. Whereas if you look back at the embryos, there's a real kind of diversity of alleles that we saw. Here, for instance, blood, again, 99% of blood could be composed of these 5 alleles. And we wanted to know, is this kind of common across the organs? Again, brain, the diversity is higher, but again, on the log scale, the top alleles in the brain really make up the vast majority of what we recovered. This has precedence in the literature. People have looked at, for instance, cardiomyocyte development in zebrafish heart. And this is with a rainbow system where colors here represent different lineages. And what you can see is at earlier stages of development, there's real diversity of cardiomyocyte lineages. But as time progresses, specific clones seem to have taken over. And on the right here, you can actually see the heart is composed of almost just 2 lineages at this point. And again, a recent work has shown this in the blood, or sorry, in the muscle of zebrafish, where there's really kind of, I hate to say clonal dominance, but there's certainly specific clones that have taken over vast majorities of the muscle space within zebrafish. So one quick way to look at this is just to put all of these alleles on a cumulative density plot. And here I'm just showing the blood. And you can see by the first 5 alleles that over 99% of the blood is composed of these 5 alleles. You can look at something like the posterior intestine, where the first 25 alleles make up almost all of what we've recovered. Again, brain, our most diverse organ, where it really takes maybe outwards of 50 alleles to get to over 90%. But you can see a really striking and similar pattern across all of the organs that we sample, where very few alleles make up the vast majority of the organism. So it's fun to kind of speculate on what that could be. Even if you put it all together, the whole zebrafish, you see this real constriction of allelic diversity. And if you compare that with earlier in development, the embryos that we captured, you see a very different pattern. You see much greater allelic diversity. So it's really interesting to think about what bottlenecks and what cell competitions these cells went through to really constrain the amount of allelic diversity that we're seeing here. So that is interesting, but can we do the same process we did before with tree building? Can we take these 200,000 cells and the 1,700 alleles, combine them with something like camel-local parsimony and build a tree of global development in zebrafish? And of course, as the setup with that kind of setup, the answer is yes. We can take here, we've actually subsetted to the 601 alleles where we have 5 or more cells, mostly just to kind of constrain the tree here so that we can actually show it. And it's already kind of stretching the limits of what I can show you. So I'll just show it to you in a video, so we'll see how this goes. And as you can kind of see this scroll by, you can see these really kind of defining edits in individual subclades, and then you can see this allelic diversity build on top of that within individual clades. And I think you can also just see the kind of scale of what we're able to recover with this system. And one of the last things that we want to do is say, since we have these diverse organ types, can we take what we know about zebrafish development and organism development in general with germ layers and ask, do each of these clades reconstruct what we know about development? So for instance, if we take that upper right-hand clade and we've labeled it here in orange, can we look at the cell types that this clade eventually leads to and say, does this actually make sense in the kind of constraints of development? So the top of this individual clade, labeled here with the number 1, is what we'll call a pluripotent progenitor. It leads to all of the organ types that we've sampled in the fish. So at some point, a cell that led to all of these diverse organ types had this single compound deletion here at the 5th to the 7th target. And so what we can do is, and you can see that clearly on the top where this deletion goes down the whole tree, and we can label on additional edits. In this case, in the middle here, another edit at the last 3 targets that then subsets it down to just the brain and the eye. And we can go throughout this whole subtree here, adding on different insertions and deletions, and constrain it down to specific germ layers and then specific even organ types. So if you look in the middle tree here, you can see on the 3rd round of editing, we're actually just leading almost equally to the left and right eye in that little green circle. So the system does seem to recapitulate known development and known germ layer relationships. But what we really like to do is link this to single cell data, right? Because we'd really like to know what each of those individual cells is. And this is again done with the Shear Lab, and this project is led by Bushra. And what they did is they took 23-day post-fertilization zebrafish, dissected out the brain, dissociated that to single cells, and put that through a single-cell RNA-seq protocol. And they were able to recover 66,000 cells, representing kind of the full complement of neuronal subtypes, from cerebellum to the optical tectum, really kind of a great single-cell data set. But what we wanted to do, right, is we want to learn more about biology here. And you can learn some of this from the single-cell data here. For instance, you can find oligodendrocyte precursor cell subtypes that we hadn't known about before. But what we really wanted to do was layer lineage information on top of this. So at the same time that single cell data was being collected, we're also collecting lineage information. And the system here is slightly different than what we used before. We've kind of split the approach into a 2 time point system where we have editing in this first 4 targets by injection at the single cell level, the single zygote. And then we have a later time point where heat shock is applied, and that induces editing at the last, at the targets 5 through 9. And when we collected that data, we want to ask very simple questions like we did before. Can we separate out different parts of the brain using these barcodes? And if you look here at the plot in the lower left-hand corner, what you can see is that the barcode really nicely differentiates different physical sections of the brain, the forebrain, midbrain, and hindbrain. So it at least is capturing at the level of the full brain. But what happens if we kind of, and what happens when we use this system where we have editing late and early? So if you looked at just the early events, we'd end up with about 4,000 unique barcodes. But when you add the early and late, the injection and the heat shock, we're getting upwards of 12,000 unique barcodes in this system. And if you compare that with what we did in the first effort, this is almost an order of magnitude better. So there are methods here to spread out the information recording out over time. And we're taking kind of this same approach forward and really trying to spread this out over development. Well, what can we do with this data set? Well, we can do something very similar to what we did before. We can build a tree with Camel-Sokol parsimony. But one of the nice things we can do here is that we can actually not just have these barcodes and the relationship between cells, but we can actually annotate them with the cell types. So here in the plot, we can add this cell type column. And here we're labeling, for instance, the forebrain, midbrain, and hindbrain, blood and progenitor cells. So we can actually add annotations to what each of these cell types are. And so one of the kind of easy questions to ask here is, do we see the same kind of clonal restriction that we saw before in the adult zebrafish that we sampled? And what I'll show you here is a tree where we label the individual internal nodes with either a black dot if they were generated in the first 4 targets with the injection, or a red dot if they're labeled with information from the heat shock. And what you would see in our original approach is just individual gray boxes for cells, right? Because we know nothing about the individual cell types here. But what we're able to do then with single cell RNA sequencing is to say, what are each of these individual cells? And so, for instance, here we have radioglia, optical tectum labeled, the forebrain. And we can look at that same question that we had before about how bottlenecked are these individual expansions. And so, for instance, here in the forebrain, which represents a large number of the cells that we captured, you can see this real expansion after that heat shock period. So the group here highlighted expands after 2 edits in the heat shock period. So there's this real kind of clonal expansion in many of these forebrain populations that is kind of a really interesting thing that we could study. And again, you see this too in kind of the diversity of types. So this midbrain and optical tectum expansion here, we see after this heat shock period as well. And of course, this just brings up a huge number of questions about what are the forces here? What's driving this? Is it selection? Is it clonal competition? Is it just kind of the normal developmental program? So this data will really allow us to kind of peek into those questions. So I just want to quickly highlight what I told you about. So we have the system for recording lineage that we kind of showed work in cell culture. We expanded it out to hundreds of thousands of cells in zebrafish development. And we're beginning to link this to single cell state in the form of, in this case, transcriptional readout, though, of course, it's not limited to that. And there's a number of different ways you can start thinking about taking this and certainly expanding it with technologies that, like what Miriam talked about last week. We can go into different model organisms. An interest of mine is taking this into somatic variation in cancer. There's an enormous space for computational improvements, combining it with methods that reconstruct from RNA-seq data, as well as even this week, new methods for inferring lineage from mitochondrial data. there's going to be a really interesting convergence of all of this. And lastly, a huge focus of the community right now is developing cell atlas efforts. So just to kind of highlight some of the work we're doing with this, you can imagine expanding this to record all kinds of different things, from transcription factor activity and development to single-cell regulatory landscapes with ATAC-seq, this RNA-seq single-cell transcriptional data, and spatial information to really reconstruct, what is happening in development. And we've taken this on in flies, and we're actually getting somewhat closer now to being able to build lineage maps of whole organisms beyond C. elegans, which is really exciting throughout development. And as I said before, one of the interests I really have is taking this into somatic variation. So you can imagine introducing this into something like a tumor, and looking at both the tumor development and combining this, say, with a mouse that records development as a whole, and understanding the relationship between tumor evolution and organismal evolution. And of course, this can be combined with whole genome sequencing methods, that's like, for instance, what's coming out of the Sanger, which will be really interesting. And lastly, we're building this with camel-stochel parsimony. It's just a pretty naive approach to take to all of this. There's an enormous space for computational improvements on tree building with data like this, and combining it with things like RNA-seq, as well as, for instance, that mitochondrial lineage tracing, to really build a rich and informative tree of development. And I think this community especially should be really excited about this, because all of the kind of stored knowledge in phylogenetics is really going to find a new home in this space, and it's really an opportunity for people in this community to lead that effort. And lastly, this is, as I said, a real opportunity to combine this with an enormous number of atlas efforts that are coming out right now. So there's just a number of groups developing single-cell atlases of development and disease, and even if it isn't this kind of CRISPR-Cas9 barcoding, there's just an enormous opportunity to be involved in the computational reconstruction of these kind of transcriptional states and their relationship throughout development, which would be really exciting. And lastly, I just want to say that this kind of fits in with our picture of development over time, of our understanding of life. We have this, early models of trees of organismal development, and of species development over time. We have this kind of trees of pedigrees of families, and what we're really hoping to do is expand this into individual development. So how does one organism go from a single cell out to the trillions of cells that each of us have, which is just a really, really exciting question. So with that, I'd like to thank everyone who's been involved in this effort. Greg and Jamie, who were involved in the first effort, and Jamie has his lab now at the University of Utah, which is worth checking out if you're interested in this. Bushra and the Shear Lab, who led the single cell effort. Marshall Horowitz here at UW, who's been instrumental in this work. The Shear Lab, and my mentor, Jay Shanduri, who's just an absolutely wonderful person and an amazing scientist. So if you are looking for a great lab, that's a wonderful home. And all of the Shanduri Lab in general, and of the people who have bravely funded this effort, especially before it produced any results. So yeah, thank you. And this has been really fun to talk.
Thank you very much. Well, that was awesome. I know something about that story already, so I didn't ask as many questions as maybe I should. But I mean, the clonal dominance thing really stands out. I mean, you talked a little bit about it, but surely you can speculate a little bit more about where does this come from? I mean, do you really think there's competition, or that some cell lineages are sort of programmed to step back?
Yeah, and I think it's a super interesting question, and I think why we haven't speculated wildly about this is because we just don't know, and this data isn't quite rich enough, I think, to make definitive conclusions. But there's just more and more examples of clonal dominance being a factor within normal development. And I mean, you can look, or even just survivor bias, right? There's a great example of in genome research a couple years ago of sequencing of the blood a 112-year-old woman, and there's just 2 progenitor populations left in her blood, generating all of the red blood cells.
And how can they tell that?
So they're looking at somatic mutations in that case. And so, yeah, it's a really interesting question as to what are the forces, right? Is it competition? Is it just is it the survivor bias? It's fascinating. And we just, especially with now the single cell data coming on board, I think it's going to be a time that we can start really answering some of those questions. But yeah, that's kind of left to the reader. I think that's what the next 10 years is really going to be interesting for.
Yeah, I mean, it's funny because it's sort of like when I think of cancer, I think of like a competitive process that's gone out of whack, but I guess maybe competition is just the rule in organisms, period.
Yeah, and I think that's the tough question, right? Is like, it's certainly cooperative, right? But how much selective pressure is there for one cell to succeed at something? And how much kind of buffering does the body or does development do in terms of making a cell population and then selecting a winner? It's a really interesting question, and I don't think we know at this point, but I think it informs how we tackle these problems too. And I think one of the challenges with an approach like this is that in all approaches like this, is that we really only see cells that survive this process. And in the single cell RNA-seq level, I think that's even more important. But we're only really able to make inferences on what survived, and there's really interesting questions about what didn't. So yeah, I think there's a lot of space for computational improvements here and inference on this data.
So we know that the nematode program is quite consistent. I mean, have you sequenced any other organisms sufficiently often to be able to answer that sort of thing?
Yeah, that's a great question. We haven't, and I think Alex coined a term for this, and I'll probably mangle it, but statistical development. What we'd really like to do is model both the kind of developmental trajectory, but also the uncertainty in that process, and the kind of variance in development. And I think we just need a lot more data to do that. But that's, I think, one of the really cool questions as you leap from deterministic development in C. elegans to what we believe is a much less determined developmental pattern in complex vertebrate life.
Right, but I mean, even like what? Yeah, I mean, where's the change point? I mean,
yeah, and I do you mean in the sense of like which species breaks it, or?
Yeah, you wonder. I mean, it's a lot simpler than a fly.
Oh yeah, yes, much simpler. And is it a spectrum? Is it, are there aspects of development that are pretty deterministic, and are there variable parts? It's really open, and especially at this whole organism level, which is, I don't know, it's going to be fascinating.
And you can sort of imagine that there might be evolutionary forces guiding that even. I mean, maybe it's sort of not advantageous for the survival of a species to have it be completely deterministic.
Yeah. Oh, and you can look at big phenotypic differences like wound repair, where there are certain species, higher order species, that do wound repair, perfectly, and at the sacrifice maybe of cancer prevalence, versus something like humans, where we scar, but we take that versus allowing cells to be more plastic in their development. So yeah, there's just a huge number of potential avenues for this.
Wow, that's fascinating. I'd like to know more about that trade-off.
Yeah, and I don't think we know. And then there's really interesting stories of like the spiny mouse, where there's just really new phenotypes that we're discovering, where these mice can regenerate a huge percentage of their skin without scars, that we had never seen before, really, in mammalian tree. So yeah, it's there's just
they are more susceptible to cancer, you're saying, basically.
I don't know in spiny mouse, but there's got to be trade-off somewhere, right? So yeah, no, there's I think an enormous number of questions that you can start thinking about answering, and I think a lot of that's waiting on both the experimental, but as well the computational tooling to start tackling these questions. And this group is it. People who are listening to this are really in the space to answer those questions.
Oh, well, it's awesome that you were able to speak. So speaking of which, it does, I mean, I like the emphasis that you put on the combination of the RNA-seq trees and the Gestalt trees, and it makes me wonder, I mean, it seems like a really natural way to benchmark the RNA-seq inferences, though. I mean, would they, if you sort of try to set up a benchmark data set and tried lots of different methods, would they say, oh, there's something wrong with the Gestalt trees? Or do you think that they would accept that as sort of like a useful, it wouldn't be maybe, yeah, it wouldn't give you the same information, but
yeah, I think the challenge too is that they're kind of converging from different ends, right? Where the RNA-seq world, the inference is more on like terminal differentiation, where we can see changes, say in terminal differentiation in the lung, where alveoli become two different cell types. In the lineage, this kind of barcoding lineage approaches go from the other perspective, where they can say things about big swaths of organismal development right now. But I think what we're really waiting for is both, just like Miriam last week, techniques that kind of can reach up the tree of development on the RNA-seq side, and then resolution of these barcoding systems to get to the point where they're saying things about more at the reliably saying things at the individual cell level. So that convergence is going to be really interesting. But yeah, no, I think it totally is a time where we could take RNA-seq and these lineage trees and converge them and see how well they agree.
That's cool. So it sounded like you said that basically flies were like a huge task to get going. And so that was like somehow flies are a lot harder than fish?
It's just experimental details on flies are just tricky. They're small, they're hard, their cells are proportionally smaller in a lot of cases, and their nuclei are a lot smaller. It lends itself to a lot of experimental challenges that we've been working on overcoming.
So I guess the choice of zebrafish early on was sort of quite a good one.
Yeah, we really got lucky there, which is great. I wouldn't be telling this story if we hadn't got lucky.
Awesome. Well, that concludes our session of 3 talks. And the next set of talks is going to be about sort of branch-specific diversification stuff. Like fitness and speciation rates and things like that. So I'm excited about that. Thank you very much, Aaron.
Well, thank you for having me.